Medicine has come a long way. Thanks to recent advancements, we're able to cure, treat, and even prevent many diseases that would otherwise stop you from seeing your 30th birthday. But despite lots of research, there are still some that don't perform quite as well as what a lot of experts would expect them to. But we're scientists. We don't dwell on the negative. We want to focus on the positive. And I'm positive that you'll enjoy this paper from the University of British Columbia called Liposomal Lipid Nanoparticles for Extrahepatic Delivery of mRNA. They designed a new type of nanoparticle that helps to avoid the liver and allow treatments to get where they need to go. And at Brain Rot U, we want to make sure that all of our students understand what all of these words mean and why this paper is so significant, which is why you'll get a brief explanation of this paper from me and from our good friends over at pdftobrainrot.org. They transform regular, dense scientific papers into a form factor that everyone can understand. A Subway Surfers clip over an explanation of this paper with the words sigma, skibbity, and goon used an uncomfortable amount of times. We'll watch this brain rot slop together and see if it could actually explain the science properly, and maybe correct it along the way. So without further ado, class is in session. PDF to Brain Rot AI. Turn boring PDFs into brain rot. Tired of dull textbooks? Transform any PDF into entertaining, TikTok-inspired brain rot content. Learn faster, remember longer, and have fun doing it. So as always, we're going to take the link from this paper, put it into this site. This site will make brain rot AI slop. We're going to watch it together for the first time, uninterrupted, see how it feels. Then we're going to watch it a second time and critique it along the way to see if it did a good job explaining the main findings of this paper. If we're lucky, the first video will be so bad that we'll have to upload it again. Okay, what do we got? Liposomal lipid nanoparticles for mRNA delivery. Okay, remember, the paper is actually called Liposomal lipid nanoparticles for extrahepatic delivery of mRNA. All right, here we go.
Liposomal lipid nanoparticles for mRNA delivery. Skibidivides are strong with these liposomal lipid nanoparticles. Goon mode activated for extrahepatic mRNA delivery. The study dives deep, showcasing how high bilayer lipid ratios rocket mRNA encapsulation to the edge of efficiency, reaching 90 to 100%. With a dank mix of egg SPH, ingo, myelin, and cholesterol, these LNPs are low-key the sigma grinds of drug delivery. They flaunt a solid core and lipid bilayer, ensuring long circulation lifetimes, outpacing on PAT-RO-like systems. This research is a ship sailing through the future of trans-FECTO and competent technology, bringing mRNA to where it needs to be.
Okay. First impression, I didn't like it. First impression, I didn't like it at all. Not that it was wrong, that was super chunky. Okay. Again.
Goon mode activated for extrahepatic mRNA delivery.
Okay. Goon mode activated. That's funny. Extrahepatic delivery of mRNA is just trying to say that they're delivering mRNA, which is the payload, that is the medicine, to anything outside of the liver. Hold up. Let's back it up for a second. We're gonna go over some of the main background of this paper, look to see what kind of problems these authors were gonna solve and how they thought to solve it. This paper comes in the subsection of the field of biomedical engineering known as drug delivery. In short, drug delivery is this field where you can make little materials that make pharmaceutical drugs better. This can mean anything from making the drug better at killing damaged or diseased cells, or better at preventing side effects. We make things called nanoparticles. These are materials that are super tiny, about 1 to 10,000 times smaller than the thickness of the hair on your head. We can then place a pharmaceutical drug into these nanoparticles. Because these particles are made of a specific size and chemistry, they can bring these pharmaceutical drugs all the way to the diseased tissue while avoiding healthy cells. These nanoparticles act like an Uber driver, delivering you to your location while making sure that you and everyone else around you are still safe. And back in the late 1900s, nanoparticles were super popular because they showed to do exactly this. The problem is that since then, there's been barely any progress. For example, only 5 nanoparticle-based treatments have been approved for cancer treatment by the FDA. This is especially bad compared to other novel anti-cancer treatments. Antibody drug conjugates have seen almost 3 times as many FDA approvals in the same amount of time. But why are nanoparticles so bad after having such a hot start? The main theory, they don't actually get to the tissue that they're trying to treat. Instead, most nanoparticles get trapped in the liver. According to some experts, more than 90% of nanoparticles that get administered to a a person get trapped in the liver within 1 hour. The liver, which is primarily in charge of clearing your blood and making sure everything looks healthy in there, alters the nanoparticles in a way that makes them unable to reach the diseased tissue. The liver's trying to help, but in this case, it's making it worse. So as you might expect, one of the hottest topics in this field of research is to design nanoparticles that can avoid the liver and instead increase the number of nanoparticles that are delivered to this diseased tissue. Okay, now back to the good part.
Showcasing how high bilayer lipid ratios rocket mRNA encapsulation to the edge of efficiency, reaching 90 to 100%.
They're using numbers and data that highlights good things from the study, but it's just kind of sticks out like a sore thumb. Not only because it doesn't have a great flow, but this is basically just saying that 90 to 100% of the mRNA that you start with, that you're trying to put in the nanoparticle, actually gets into the nanoparticle. And that's very impressive. That's good, but it's also pretty standard. That's not like a unique thing to this specific new nanoparticle that they're making. Where they almost do a good job is talking about these bilayer lipid ratios. But first we need to talk about lipid nanoparticles, or LNPs, and what they're made out of. As you might have guessed, they're a nanoparticle made up of several different lipids, which are shown here. An ionizable lipid, a helper lipid, a stabilizer, and finally, cholesterol. These lipids usually need to be added together in very specific ratios in order to make a pretty little particle. So for example, a typical LNP, such as the one used in the COVID vaccine, is about 50% ionizable lipid, and only about 10% of it is helper lipid. But what if you didn't follow these guidelines? Well, that's exactly what these authors did. They drastically reduced the ionizable lipid ratio while raising the amount of helper lipids. They wanted to see if using all of the same components but changing their relative amounts would change how this nanoparticle looks. And in turn, if this different looking nanoparticle had a different function. If you can change the function, then maybe you can avoid that pesky little liver. So the brain rot actually does a good job here, talking about those bilayer lipid ratios. But then they do something that's not so great.
Dank mix of egg, SPH, ingo, myelin, and cholesterol.
Christ, oh, that's tough. I'm not going to pretend like brain rot is ever going to understand or know what sphingomyelin is, right? I get it. They can't say the name of the lipid, but spelling it out just, ooh, that's just not nice to the ears.
LNPs are low-key the sigma grinds out of drug delivery. They flaunt a solid core and lipid bilayer.
This is referencing what the final structure of both the original LNPs and these new LNPs looked like under an electron microscope. When they used a small amount of helper lipid, it looks like a regular LNP, just a jumble of lipids with a single lipid layer on the outside. Electron microscopy can be a little confusing, but that's what these little gray droplets look like on this left-hand side image. Now let's look at the right image where they increase the amount of that helper lipid. You'll notice that a lipid bilayer, or two distinct layers of lipids, form around that initial LNP. This is shown on the microscopy by that darker, smaller droplet inside of the larger, lighter droplet. They called this new structure a liposomal LNP, aka one fatty particle giving a nice big hug to a smaller fatty particle.
Ensuring long circulation lifetimes. Outpacing on Patro-like systems.
Another example of doing really well until it doesn't, until it loses beam. Unless you read this thing, what the fuck is Onpattro? It's commercialized nanoparticle treatment that also acts in the liver, which is exactly what their LNPs are trying to avoid. It's a really good standard, it's a really good control. To test this, they injected mice with colorful versions of either type of nanoparticle. Based on how colorful each mouse organ was, they could determine where each type of nanoparticle was going. The normal LNPs, on the top, were very localized to the liver, but the liposomal LNPs, on the bottom, were much more spread out, meaning that they were likely able to avoid the liver and get to different organs. In fact, when they crunched the numbers, they saw that liposomal LNPs got to the spleen 50 times more, and into major lymph nodes 150 times more than regular LNPs. And apparently it wasn't statistically significant, but it looks like liposomal LNPs were really promising at finding the pancreas as well. By having this liposomal bilayer on the outside with the LNP on the inside, this improves how long these nanoparticles are circulating through your body without getting trapped in the liver. And that's good. By not getting trapped in the liver, you give these nanoparticles extra time to get to the organs or the tissues that they are trying to get to. And the brain rot is trying to say, like, yeah, look, their new design is better than Onpattro. But they don't explain what Onpattro is. So if you're just scrolling through this for the first time, you're like, what the hell is this? They also don't explain what the structure is. How nice would this be if just really quickly, they just showed like a little picture of just like, hey, here's what their liposomal LNP looks like. They have the figures in here. Why not just show them? That would be awesome. Just, I don't know. Okay, overall thoughts on this video, like, there's nothing particularly wrong, but this is probably the worst one I've seen so far. Not in terms of accuracy, but in terms of enjoyability for someone who didn't already read this. I'm not showing this to people to help explain what this paper is and how cool this is. I'm not showing this to my mom. Say a bunch of things, but don't draw any connections to them. It just feels like a bunch of random words just spat at you. So because this was so terrible, now you know what time it is. It's time for another video. Let's get it. So now we have lipid nanoparticles for mRNA delivery. This is the most like ChatGPT title of all time. Like probably the most Google search thing if you looked up the words lipid or nanoparticles or mRNA in any case in the last 5 years. This is probably what you would get.
Lipid nanoparticles for mRNA delivery. Ski bit I vibes in the lab as researchers goon hard on LNP mRNA systems. These liposomal lipid nanoparticles are low-key the sigma of drug delivery, achieving extrahepatic delivery with high bilayer lipid ratios. Picture this. A dank liposomal structure with a solid core, rocking a for 0.67 lipid ratio that yields 90 to 100% mRNA encapsulation. These LNPs are the edge of trans-FECTO and competent tech, ensuring that mRNA gets where it needs to go. Long circulation lifetimes make these bad boys the ultimate ship, ready to revolutionize gene delivery. It is a game changer.
I mean, it made me laugh, so it can't be that bad. Let's break it down real quick.
Lipid nanoparticles for mRNA.
First of all, lipid is just a regular word. How can it not say lipid?
Ski bit I vibes in the lab as researchers goon hard on LNP mRNA systems.
Goon hard on LNP systems. Amazing. Thank you, pdf@brainrot.org. I'm sorry for talking about you.
Picture this.
Picture this.
A dank liposomal structure with a solid core.
Boom. Boom. This is the single best 4 words of all of the brain rot we've seen today. They're actually trying to describe what the hell's going on. Picture this, a dank liposomal structure with solid core. Beautiful. That's excellent. But then watch what happens.
Rocking a for 0.67 lipid ratio that yields 90 to 100% mRNA encapsulation.
That is just a lot of chunky numbers.
4-0.67.
Again, I'm coming back to if I'm scrolling and I'm seeing this, I'm out. I'm so out. I don't know. Am I being too harsh on this? Like, am I being too harsh because this is like my field? I don't like that they didn't include the parts that I think are coolest. Am I being too harsh?
Long circulation lifetimes make these bad boys the ultimate ship.
Good. Like, they are finally explaining why, but it just like it came after such a large explanation that I thought was not well linked. You know, when I was reading this, I thought the circulation lifetimes part, the structure of the LNPs, and where are those LNPs going, those are the main points. Like the fact that it's 50 times more going to one part of the body and 150 times more to another compared to regular LNPs, that's really important. And I wonder if the AI is trained to say, hey, numbers usually means good, and so highlight those, like emphasize those. So maybe it's not the brain rot's fault that the abstract didn't have those numbers to highlight it. Anyways, I'm not here to critique the authors. I'm here to critique this stupid website.
Revolutionize gene delivery. It is a game changer.
That's another little slap on the wrist. It says it's a game changer. That didn't seem like a game changer because they didn't really compare it to anything else. They just said longer circulation times. And in the last video, they did mention it and did compare it to Patreon. I didn't like that either. So maybe it's me. So for all this, I think we have a Brain Rot University first. Both videos were trash. I did not like either of them. Other times I've used this site, it seemed like the first video that was made was usually pretty good, but kind of rough. And the second was usually way, way better. Like there was a lot more useful data and the explanations were a little more clear. Whereas, yeah, this one was just, they both kind of sucked. Maybe I'm grumpy, but I think there was a lot to be desired for both. And I don't think either of these videos I would have watched while randomly scrolling through TikTok and would have gotten something out of it had I not already known what this paper was about. If the whole point of this site is to help you enjoy studying more and learning these new things, it's not really doing its job. Now, it also all comes back to how hard is this paper, right? Like the most successful time we've used this site has been when there was a relatively straightforward paper, a lot of weed searching that's going on in this paper, to be fair, right? There's just like a lot of numbers, right, that are just saying, hey, these are the different ratios that we use, like how much of each component of those LNPs did we use. And I wouldn't be surprised if the brain rot site had a hard time parsing through all of these data. So we know the site draws heavily from the abstract. The abstract doesn't really give it a whole lot to play around with. So I guess it's just doing the best that it can, but yeah, rather than past, I wouldn't like or watch any of these. That doesn't matter, because it's not about what I think, it's about what you think. So let me know down below, who would you like better? Massey PhD, Professor Brainrot AI Slop, either version 1 or version 2. Am I being too hard on them? I don't know. You tell me. You can also let me know what you like to see from the series, any other papers that you'd like for me to break down, or to let the Brainrot break down. If you learned something today, be sure to give this video a like. And if you learned a lot of things today, make sure you subscribe. You can also comment over to Instagram. That's where most of our community hangs out and chats about all things nanoparticles and biomedical engineering. See you later, nerds.